presentation will appear soon. Meanwhile, I will tell about myself a few words. Basically, I was invited by the Distinction and Creativity Agency, and as I understand, Distinction and Creativity Agencies, first of all, involved in education process. Basically, agency is trying to bring to the Syria best of knowledge, build a scientific community via integration with different countries, sending their own students to the different countries, for example, to the Russia. And I actually, in my lab, are hosting 4 of them. So basically, later on, you will be, maybe on the day 2nd and 3rd, you will be having presentation from them. And today I will be briefly presenting what we are actually doing in our laboratories, in our centers. And I mainly would like to target all of you, the students, the PhD students, actually in a recruitment matter. So basically, I will be telling you what kind of projects we are doing and what you are potentially might be doing in my laboratory. And since I am head of several centers, so basically I will be briefly discussing what you can do not only in my laboratories but laboratories of my centers.
Excuse me, Dr. Pavel, you have 15 minutes.
I will try to be brief. So basically we are growing quite fast. I represent a person who actually returned from United States. I spent there 10 years and we are growing fast. So basically I started from the several people. Now we are more than 100 scientists in several laboratories. Basically what I'm going to talk about today, it's about inherited diseases, so genetic disorders. As you may know, it's more than 6,000 genetic disorders. So basically, almost all of us who is present in this particular room are carriers of particular genetic disorder mutation, maybe several of them. But obviously, it's a rare event when autosomal recessive disorders actually happening. But if you sum of them, it's actually not that rare. It's almost like 2 to 5% in our population who actually suffer from genetic disorder. And what we are doing different instruments, platform, and actually genetic therapies for genetic disorders. So basically, it's a simplified scheme how we develop therapy. Basically, you design your vehicle. In our case, it's mostly AAV, adeno-associated virus, which we deliver. And then you are doing basically proof of concept. You are choosing proper animal model and trying to treat this particular animal model. In most cases, it's mouse. So basically, we are trying to cure mice. And then you are going, if everything's successful, you are going to the clinical trials. So as an example of what we are doing, for example, it's KCNV2 gene therapy. It's a disorder which actually happening at early stage. Basically, manifestation is happening around like 2-3 years old kids. And they're losing their vision because photoreceptor cells are dying, constantly dying. And so basically KCNV2, it's a part of potassium channel which actually involved in regulation of this particular channel. And without this channel, unfortunately, photoreceptors quite dysfunctional, and actually it also limits the half-life of these particular receptors. So basically, we are very close to the clinical stage for this particular genetic disorder, and hopefully we will be like in 2 years in clinical trials with this particular one. The other one, it's congenital adrenal hyperplasia. It's actually very, very frequent disorder. It's actually a rare genetic disorder, but from all rare genetic disorders, it's one of the most frequent. So basically, the frequency of this particular disorder is 1 to 10,000. That means that carriers, it's the frequency of having a mutated allele, it's around 1 to 100. So if 100 people is sitting here, it's most likely that one of you are actually having this mutated allele in your genome. Sorry. So what we are doing a combined approach. Basically, we are trying to deliver functional gene to the adrenal glands, and at the same time, because adrenal glands is quite quickly renewing organ. So basically what we're trying to fix problem in genome. So we are actually doing genome editing. So we are actually trying to use and using successfully CRISPR-Cas instruments, which you were heard about them in the first session. So what else we are doing? Basically, we are developing platforms because all of this AAV belongs to somebody. And what we are doing, we developing our own adeno-associated viruses, which will be free of any particular pattern. So basically, we create new viruses with new features. Basically what we do, we design new one using different bioinformatic technology, structural biology, and afterwards we are testing them. I'm not going too deep in the details of how we're doing this, but it's a combination of in vitro approaches, testing, for example, if the target virus is for example, for ophthalmology for retina disorder. So we are testing on mice retina, on iPS-based retina-like structures, and also obviously on a donor retina as well. What we achieved, we actually generated several generation of viruses and quite successfully. For now, we have a collection of 9 viruses, which all of them have a slightly different trapezium, and this allows us to apply our library, AAV library for different particular genetic disorders with different trapezium, for example, liver, retina, adrenal glands, muscles, so for different kind of types of genetic disorders. And as an illustration of successful results, so basically here retina with GFP staining and recoverin at the same time. So this particular type of virus which we developed quite successfully target photoreceptors. So it's actually ideal chimeric synthetic AAV virus for targeting photoreceptor now. And we compare with all available wild type AAV viruses and synthetic viruses from different companies. Now we have best viruses which are actually targeting photoreceptors. Also, we develop in different viruses, for example, for muscles. So basically here you see in vivo images of targeting a particular muscle. It's also quite successful result. Besides gene therapy, we're actually doing a lot of autoimmune projects related to autoimmunity, like type 1 diabetes. At the same time, we are also doing research in field of endocrinology and oncology, because I'm head of Center for Personalized Medicine at National Endocrinology Center. So, but obviously nobody could stay a part of recent COVID-19 story. So we also, together with Moscow City, with Department of Healthcare, performed quite a huge cohort study, and we basically what we are trying to prove that actually it sounds a little bit funny, but at the beginning of COVID-19 story, you remember, like everybody was questioning whether immunity actually develops for COVID, for SARS-CoV-2 or not. So basically, it was a lot of studies initiated at that time. So basically, what we did, we actually proved that yes, human immune system actually developed immunity towards SARS-CoV-2. Basically, that data is published in a journal. So basically what we show that with different approaches, basically we were comparing ELISA, different kind of antibodies, ELISpot, which actually test T-cell response, specific T-cell response, flow cytometry data for different T-cells, CD4, CD8, and so on more than 10,000 people. And we actually were doing it like continuously. We were looking for following patients for several periods, at least for 270 days, actually more than 200. For now, it's more than 1 year and a half, so we were following patients. And the short answer is that yes, your immunity actually develop resistance towards SARS-CoV-2, and all of these graphs actually illustrate that antibody after first contact with virus actually develops quite well, and obviously not for everybody. Obviously, the older you are, the less less antibodies you have, usually, not always. But the black and white answer is that if you did contact with SARS-CoV-2, you do have antibodies, you do have T cells which actually stay with you and which actually limit second and maybe third infections, SARS-CoV-2 infection. And if not, but it's definitely limits the severity of the disease. I'm stopping for now, and if you have questions, I open for the for answers later on. Thank you.
شكرا كثير وبرحب كثير بالزملاء الأطباء الروس بمشاركتهم بالمؤتمر. بيسعدني أقدم الدكتورة تاتيانا شكرات، رئيسة قسم. مو بعدين المناقشة بعدين. المناقشة آخر شيء. آخر شيء أفضل.
Dr. Pavel, discussion will be after the session. يطلع معنا إنجليزي أكتر من روسي. أنا بحكي منيح بس نسيت. لأنه هو بيحكي إنجليزي بيحكي بوصلي ده. أنا بحكي منيح بس لأنه حاجة إنجليزي صار تطلع الإنجليزي لحالها.